Hey, everybody, welcome back. I'm Susan Lindner, your host of the Innovation Storytellers Show, where we get behind the innovation and talk about the story that got these amazing breakthroughs to their finish line. Very interested in my guest today because for those of you not in the biotech world or in this sphere, last week, 2 weeks ago, rather, in Boston, I'm in July at the moment. So 2 weeks ago in Boston was a big biotech conference called BIO, and it brings together some of the world's greatest scientists to talk about the breakthroughs that are happening. I didn't get there, but I got a lot of inbound requests for interviews, and I was most impressed with this inquiry I got from Dr. William Pao. I'm going to tell you a little bit about him, and then we're going to dive into his book that came out this year and really get into some incredible stories behind medical and bio breakthroughs that have been going on that all of you just need to hear about, both as inspiration on the breakthrough side and to hear the incredible stories that got them there. So, Dr. William Poe is a physician scientist whose career has spanned academia, industry, and biotech. Motivated by his father's death from cancer when he was just 13, he started his career as a thoracic oncologist, a lab researcher, and clinical investigator at Memorial Sloan Kettering Cancer Center in New York, and then Vanderbilt University in Nashville, where he also served as the division chief of hematology and oncology. Previously chief development officer for Pfizer, and prior to that, head of the Roche Pharma Research and Early Development Unit, he oversaw the discovery and development of portfolios of new molecular entities, many of which have been approved to treat a variety of diseases from cancer to rare blood disorders. He has advised numerous professional associations, served on editorial boards of prestigious medical research journals, and received multiple awards and recognitions. He is currently the CEO and co-founder of a cancer biotech company that is in stealth mode. So we're not going to talk about that, but we are going to talk about your incredible book, Breakthroughs. Dr. Powell, thank you so much for joining me.
Susan, thanks a lot. It's a pleasure to be here.
So let's dive in. Can you take us back to your start? Dr. Powell, tell us a little bit about your story and your decision to enter into this field.
Yeah, so it really starts with the unfortunate passing away of my father when I was age 13. He had metastatic colon cancer, and there weren't very many treatment options at the time. And I won't go into too much detail, but he died sort of all of a sudden. And he was a doctor. My mom was a doctor. So it was sort of expected that I would go into medicine. But at that point, I really did vow to make a difference for patients like him. Of course, at age 13, I didn't really know how I was going to do that. But subsequently, I ended up getting an MD and a PhD, doing laboratory research, internal medicine training, and medical oncology training. And after a gazillion years of training, I was then able to be a thoracic oncologist, doing research in the lab, also helping to develop new drugs in patients in the clinic and seeing patients. So there, I initially started in academia and thought that I would be satisfied. But then I helped other companies develop medicines, including Tagrisso, which is developed by AstraZeneca, which is used for lung cancer patients. And then when the door came knocking, I got headhunted and asked to head up Roche Oncology drug development for the European-based part of Roche, which is called PRED, or Pharma Research Early Development. So sort of an opportunity I couldn't refuse. So I decided to do it full time, and I have no regrets, and then have been able to develop medicines in oncology. Eventually, I became head of that whole unit, which was immunology, infectious diseases, ophthalmology, rare diseases, and oncology, and heme disorders and other indications. And so I was able to have an impact in multiple different disease types. And then ultimately, I was the chief development officer at Pfizer, as you mentioned as well. So it's been a very fulfilling career. And the reason I decided to make the plunge, some people say going from the academia to the dark side, was really to, have an impact on multiple patients. As a physician, I could have individual patients, which I really valued. But when you develop a medicine that treats many patients, then you can really have an impact across the globe.
Yeah. You and I share a background. Certainly, I'm not a physician, but my first job out of college was at Memorial Sloan Kettering Cancer Center. I think it's an incredible place for anyone to start their career. The commitment to excellence is so high. And my last gig after 10 years in public health was as a field epidemiologist working through the Centers for Disease Control, tracking variant and resistant strains of HIV. And so I would spend my days as a field epidemiologist through the New York City Department of Health, going to hospitals all over the city, interviewing patients. I'm an anthropologist by training, interviewing patients about all of the sex and drug injecting partners they'd had since 1970, collecting those people's names, and then following up to track the spread of variant strains of HIV coming into New York City, and doing that in English and in Spanish. So very varied population across 5 different boroughs and collecting that data. You have written a book about breakthroughs. And, this book, it strikes me, we were so craving breakthroughs in HIV in the 90s. I started by working in brothels in Thailand, doing HIV education with sex workers and their customers in Thai. And then, came back to do this HIV work in the field in my hometown. But we were so aching for a breakthrough that went beyond the condom. And I remember when the first, AZT trials hit, and those shifts, and just how incredibly challenging they were to keep people on, those types of studies, because the medicines felt so violent in the body, right? I mean, it was combating a horrific disease, but boy. And then to think about where we are today, most people think about, HIV now as a treatable disease, whereas it used to be a death sentence. I remember patients cashing in on their life insurance policies, just to be able to use them before they passed. Like it was serious. You've chronicled some incredible breakthroughs and the stories behind these breakthroughs. And I'm wondering if you can take us on a little tour. I was especially interested in, the HIV related stories. Can you give my listeners, an inkling about some of that work and the stories behind that?
Yeah. So first of all, your career sounds really remarkable. And so, yeah, I mean, I also lived through that period, and actually I was an intern, and I remember patients coming to the hospital really with, horrible infectious diseases as well as cancer that was developing in patients with HIV, and there was really no treatment. But thanks to medical breakthroughs, there were multiple antiretroviral therapies that were developed. And as you mentioned, today it's really a chronic disease. Unfortunately, a lot of patients are infected around the globe, and it's not cured yet, but it is a chronic disease and manageable. Now, for breakthrough, my book, I wrote about 8 different molecules from 8 different companies. And I wrote the book for several reasons. One is I really wanted to highlight the unsung heroes of drug development. Multiple times they're very invisible. They're working really hard. They may not be getting publications, as you might get in academia. They're not out there in the public, known very well. And they're actually not, cashing out from, huge multiples in their companies, because they're just working every day really hard to try to make a difference for patients. So that was one reason. The second one is there are not enough books about innovation in drug discovery. There's a lot of books about innovation in other fields, but not drug discovery. And then the third was to inspire the next generation of scientists and others to make breakthroughs in their own areas. One of the book chapters is about lenacapavir, which is a new medicine. It's a capsid inhibitor, which I'll explain in a second, for patients with HIV. And actually, it just won the Science Magazine 2024 Breakthrough of the Year award because it's given twice a year and basically can really suppress HIV significantly, currently on top of antiretroviral therapy. But now it's even potentially going to be used to prevent infection, meaning you could get a shot and then prevent infection from even happening.
Wow.
So it's really quite remarkable. The story starts very interestingly, basically from a scientist named Wes Sundquist, who was not necessarily interested in making a drug per se, but really was fascinated by capsid structure and capsid biology. Now, HIV is a virus, and like other viruses, it has basically a capsid, which is the shell of the virus that includes and shields the RNA and other proteins from the external environment. And most people hadn't even thought about the capsid as a place that could be a target for drug therapy. Most people were thinking about, the enzymes in the actual HIV virus that are necessary for replication. Enzymes are much easier to drug than structures. But coming back to Wes Sundquist, then basically he was just fascinated by capsid biology, and through basic science started to understand what were the structures important for the capsid, how did it form, and then eventually fell onto the discovery that if he put mutations into that capsid, you could actually affect the life cycle of the virus. So that was really significant because then it suggested that this capsid could actually impact whether the virus lived or not. And that was the insight then that Gilead scientists had. Hey, maybe I could actually make a drug out of this. But that took a long time. It took a lot of persistence. Like any molecule that's ever been developed and approved, the scientists have to work resiliently with persistence, a lot of ups and downs. You're really exploring the unknown here and trying to turn something that's undruggable into something that's druggable. I could go on the story, but it's really a fascinating story that starts from true basic science inquiries, which are really important for us to do because we never know where they're going to lead.
And this is true of any innovation, right? We don't know when we start off, and we have to be open to the risk associated with that, right? That we might go down a rabbit hole or we might strike gold. And part of the innovation process is giving ourselves the creative freedom, right? And the scientific rigor to be able to walk down that path.
Yes. And I give Thomas Szilard from Gilead a lot of credit. He's the one who sort of oversaw this development process. And at many times you'll see in the story, he was about to be shut down or the project was not making any progress. Maybe funds needed to be put somewhere else, but he really stuck with it. And then they were able to make this molecule that's maybe just given twice a year. There's many other issues that molecule encountered, including, the original intent was to make a molecule that's given orally once a day or twice a day.
Mm-hmm.
But it turned out when they made this molecule, it was very unconventional and it was very insoluble, meaning that you couldn't make it into an oral pill. So instead of, wringing their hands, one of the scientists there then said, well, what if we turn that to our advantage? Instead of it being a disadvantage, what if we gave it subcutaneously, meaning under the skin, and took advantage of the fact that it's not soluble, and then maybe you can only give it twice a year. So that was also another insight and breakthrough by the team there that basically made lemonade out of lemons, so to speak.
I noticed this point in your book where you say after 15 years of intense research and development, not a single bispecific antibody had been approved for use as a medicine, while monospecific antibody programs surged ahead. Research labs quietly shelved their bispecific cousins, right? So, yeah. And we hear these stories in innovation across industries all the time that we get so close, right? There's a great book called 3 Feet from Gold, where a gold miner is spending his whole life looking for gold and then stops 3 feet from the biggest vein of a lifetime. And it's only like, oh, maybe we need a different set of eyes, that the gold miner actually invites an engineer rather than his gut to begin looking at something and say, oh, by the way, just hang right here. And there's the vein waiting for you. But these kinds of things. And I also love the story in the fact that, a younger comes up to the older, maybe like tired and haggard kind of researcher who's spent his life hammering away trying to find that vein and then eventually finds it. I'm curious, there's that inflection point where you have to make the case to keep going. What is the storytelling that happens when you're in that fork in the road and there's one side that says stop and another side that says go? What is the story that gets told at that juncture? Because so many of my listeners are at that juncture.
Yeah, I mean, I think with every breakthrough, there's probably comes at least one or two really almost not life critical, but, possibly killing event that would kill that idea or kill the development of that idea. So really, first, people have to persist. They have to be resilient. They have to really understand what it is that they're trying to develop. And is it totally crazy, or is there really data to support what they want to do? I think they also have to have an idea of, what is the next so-called killer experiment that I could do to either prove my hypothesis or disprove my hypothesis. Now, killer experiment is, it's interesting, that may have a negative connotation to some people. And actually, when I was the head of Roche and I tried to introduce the concept of killer experiment, people really didn't like it. But it is a very good, useful tool for people to think about, what would be the absolute experiment that would either shut my project down or enable me to keep going. And I think the storytelling then really becomes about, here's the evidence that this is really important. This is the data on why I believe that I can achieve what I want to achieve. And these are the next series of things that will either point the way forward or, tell me to stop.
Or kill it. Yeah.
Exactly.
It's a killer experiment with that in mind.
Exactly. Yeah.
You write on page 126 in your book, this is the reality of innovation that is not widely understood. Many great ideas never make it to fruition because the functioning prototype can't be reproduced at scale efficiently enough to be cost effective, right? And there's so many layers even to that it may be a great idea, but the market won't bear it, or, and I love the way that you position this researcher as well. You talk about the researchers. Kunihiro Hattori is approaching retirement. He has a placid round face and a businesslike manner that speaks of a serious career built on long hours in the lab. But these days he prefers bird watching more than that. And so like we all know that feeling of innovation fatigue too, where you just keep running into the same wall over and over, and you think you got it, but damn it just doesn't work.
Yeah. Well, he's in the chapter about emicizumab, which, as you mentioned, it's a bispecific antibody for a blood disorder called hemophilia A. That particular project, as you see in the book chapter, took 10 years to go from an idea to eventually putting it into patients. That's a really long time. And, that project also had a timeline where the bosses were like, you have until X date to prove that this has legs, or else we're taking away all of your resources. And in fact, many of the resources were taken away already before that. So, but this person particularly had conviction and really believed, based on the data that he was generating, that it was possible. And now that medicine is approved for hemophilia A and really has made a huge impact on patients.
Yeah. There are like just so many themes across the board in innovation. I think maybe, in the biotech world, you're a little bit more generous with timelines simply because so much of the work is breakthrough from jump. Obviously you're building on, the forefathers of science that produced experiments prior, but it's always new, isn't it? I mean, you're always, even if you're rehashing something, you might have a new demographic that you're working on. You might have a new age category, gender, et cetera, that you're looking at these for. And for all of my listeners, these are the same confrontations we find all the time is, am I done with this search? And a chief innovation officer who I really enjoyed working with at Corning Glass, he always kept a slush fund around for the old ideas that never really made it, that were like still in the barn, but, they thought that horse might still have a chance to run one day. And so how do you kind of keep the home fire burning? Is that an advice to innovation portfolio managers, that they do keep a little slush fund for the old ideas to keep poking at it? When do you give up,
If I say
Dr.
something
Pro?
here, a lot of people of the executives or the strategy people might not be very happy, but because some of the projects refuse to die, right, so to speak. And then, you do need to clear out projects that really aren't going to work, because you need to have those resources and the money for the projects that are going to eventually work. But I think, it comes down to really what level of conviction do you have, and what data is possible to generate to, again, show that this idea is going to work or not. Now, what you could do, I think, in that situation is really limit the team's focus to doing that so-called killer experiment, right? You don't have to front load everything all at once and spend a ton of dollars, when really what you need to do is this one killer experiment, so to speak. And then once that's shown to be successful, then you can unleash, the rest of the funds. So I am supportive of, keeping a slush fund around, but you also have to really be disciplined, I think, and not waste money on stuff that's really not going to work. It's not a science, it's more of an art, I think, but you do have to be disciplined about it.
Yeah. And then, maybe you come back to it again.
Yeah. Well, I think some of the themes in the book you'll see are also institutional memory makes a huge difference. So, you may have failed at one period and then shelved that project. But then, some new insight comes around, or maybe there's in the case of Pfizer, which is Paxlovid for COVID, the Pfizer team had worked on the original SARS virus back in the mid 2000s. But then when the virus disappeared, they shelved all that and then got rid of the infectious disease team there, in fact. But then when COVID came around, there were enough people that had that memory that were able to revive the project, so to speak. But they had already had the substrate in order to move faster and really at pace. So there is a role for institutional memory for sure.
It's so fascinating. I just finished reading a book called Same as Ever. I'll get the author's name. But Same as Ever was an incredible book for me because it said that we are so taught in medicine and science to look for the anomalies, oftentimes rather than the pattern.
Mhm.
And as a result, we're shocked when a big, epidemic rolls around. Meanwhile, the history of humanity is punctuated by epidemics. And the author goes on to say that 100 years ago, we had 4 epidemics moving simultaneously across America and the world. The Spanish flu, measles, mumps, rubella, right? They were part of our daily lives, and it wasn't uncommon for any family to be wiped out by one or numerous pandemics simultaneously in the same house. And he actually quoted Mayor Ed Koch's communications director, who said, "We had 4 epidemics in our house. My uncle had polio, my brother got measles, my uncle got, and it was just one person after another had these diseases." So fast forward 100 years, COVID rolls around, we are in complete freak out mode, right? We have no idea how to handle this emotionally, physically, scientifically, right? It's like we lost the institutional and the human memory of what it is to experience pandemic-like event. I mean, for many people, they weren't personally touched by HIV, so they didn't know that experience. And Ebola felt like it was a million years away. In Africa, swine flu, right? Foot and mouth disease, like all of these things, they didn't seem to impact us blessedly in the United States. So, wow, we lost our brains.
Yeah, I mean, the book doesn't really go into this, but we take for granted, I believe, since the advent of antibiotics and vaccines, right, that infectious diseases are really tamed. I think you mentioned some of it, but even the generation, well, I guess more than 100 years ago, people used to have 10, 12 kids. And it wasn't uncommon that several of them died before even the age of 3 or even 1 because of infections. So antibiotics and other innovations have really made a huge difference in human health that we take for granted, I think. Yeah.
And we don't see incremental changes as innovation either. The book that I was just mentioning, Same as Ever, credits something like a 70% reduction in cardiovascular deaths over the last 100 years as a result of research, but that change is only a 1% shift year over year, right? So there's no headline that says like cardiovascular deaths down 1% again this year.
Yeah.
Like,
Yeah.
that's breaking news.
That's true.
But the number of humans actually saved in those breakthroughs could be, 250,000 people, maybe even more.
Yeah.
They're just still on the planet, so we don't notice the breakthrough.
Yeah, and then I think the other thing about medical breakthroughs, the storytelling is challenging. As you see in the chapters, right? Some of the stories span 100 years from the original description of a disease to, maybe 50 years later, someone actually figured out why that disease is happening to then, another 20 years where the molecular basis is found and so on. So, and then ultimately, many, many, many people work on a molecule to get it over the finish line. In fact, at Roche, when I counted up the number of people that worked on a molecule to, get it approved, it can be up in the thousands. And that's not unusual. I think if you ask many of the companies, it would take that many. Now in biotech, you have very few, but they don't take it over the finish line, so to speak. And so telling a story that spans 100 years with thousands of people becomes very challenging too. Because as you know, we like heroes, we like to focus on 1 or 2 people, but the innovation process in the life sciences is very different, and it really requires a team effort and can span decades.
Absolutely. And and a lot of other businesses too. I've mentioned this on the show before in other episodes, but Corning Glass keeps an innovation archive, right? They're a 150-year-old company. Maybe I could be getting that wrong. Might be 165 now, where they keep an innovation archive. And it turned out that the glass that was needed for the Moderna vaccine that had to be kept at very cold temperatures for very long periods of time was recipe for glass that was invented in the 1930s. And because they had this archive, they were able to revive that recipe. They frankly hadn't had a need for it for quite some time, and wound up manufacturing that Moderna vaccine in the only factory that was allowed to operate 24/7 in Wuhan, China during the virus, during the epidemic, to manufacture those vials. So that factory was the only one that kept going throughout the course of the pandemic, which I found fascinating.
Yeah, I didn't know that story.
Yeah. Well, coming back to your book, though, like, can you also share? I think the story is fascinating around CRISPR and exocell. Can you share a little bit about that story? Because I think it's really instructive for my listeners about one more level of storytelling around this breakthrough.
Yeah, so exocell it's the nickname for a much longer molecule name, which I can't even pronounce. But it's basically the gene editing molecule approved from Vertex and CRISPR Therapeutics for sickle cell disease, which is a rare hematologic disorder. It's a genetic disease that's caused by a specific mutation. Now, this is a story that spans more than 100 years, where the initial description of sickle cell disease occurred in the 1920s. And then basically it took till the 1950s for people, scientists, doctors, to even come up with a potential way to treat sickle cell, which is the observation that actually human beings have different forms of hemoglobin. So sickle cell is a disease that affects your hemoglobin, which is the protein that carries oxygen in the red blood cells so that we can live, basically. And by the 1950s, it was observed that we actually have different forms of hemoglobin in our bodies. There's a fetal hemoglobin, and there's a neonatal hemoglobin, a fetal hemoglobin, and then an adult hemoglobin. And it turned out that Janet Watson, who was a pediatrician in the 1950s, observed that when patients were born with sickle cell, they didn't immediately have the symptoms. And these symptoms can be really bad. They can be life-threatening. They can lead to strokes. Basically, because your red blood cells are sickled or not normal, they can clog the vessels and then cause severe pain, strokes, multiple different really devastating disorders for these patients. So she observed that basically that when patients are born with sickle cell, they don't immediately have the symptoms, and reasoned that maybe they had fetal hemoglobin around still that was not sickled, and then eventually over time you replaced it with the sickle cell hemoglobin. Now, the reason that's important is then people said, hey, maybe I could make people still have fetal hemoglobin, and then that would be a way to treat patients with sickle cell. So by the 1970s, a big scientific question, this was not a therapeutic question at the time, but a big scientific question was, how do cells switch from fetal hemoglobin to adult hemoglobin? And so scientists like Stu Orkin at Harvard, but there were many, many other scientists, started working on this problem. And starting in the 1970s, Stu's lab basically worked on this and had many, many insights in other areas, but they were never able to find how cells switch from fetal hemoglobin to adult hemoglobin. In fact, the whole field sort of gave up by the 1990s, 2000s. So even though, all these people, all these brilliant scientists were working on it, they really couldn't find any insights. So it took eventually a new MD PhD student, Vijay Sankaran, who came to Stu Orkin's lab and said, hey, I want to work on this. And even Stu Orkin was like, why do you want to work on this? Like, nobody's working on it anymore. But this person was particularly interested in it. And he even worked in Stu's lab for a couple of years and still had no insights and was not able to make any breakthroughs. Now here, what happened was then it came from left field. The real insight came from actually molecular epidemiology. There were some genome-wide association studies. These are molecular studies done in populations to understand if there's particular genetic variants associated with diseases. And I'll cut this long story short, but through a GWAS, or a genome-wide association study, a new gene called BCL11A was implicated in fetal hemoglobin switching. Now, no one had even thought about this gene being involved in this process. So the lesson here is really you have to also pay attention to other fields and not be, totally tied to your area, but be open to new other ways of studying a problem. In this particular instance, molecular epidemiologists were able to find this target. Then, to make a long story short, Stu's lab was able to show that, BCL11A indeed was the gene that was implicated in fetal hemoglobin switching. And then the question became, for a therapeutic, how do you actually make that into a therapeutic, right? And fortunately, CRISPR came around. This is not a particular target that's amenable to small molecules or large molecules, which are the conventional types of medicines that we make. But it turns out CRISPR, which is a gene editing technique, would then allow for correction of the defect that would allow then BCL11A to then be turned off so that you could actually reverse fetal hemoglobin production or keep fetal hemoglobin persistent. So in any way, the long story short is CRISPR and Vertex then were able to come up with a way to gene edit BCL11A and thereby actually even cure patients with sickle cell and also another form of a rare disease that affects hemoglobin called thalassemia. Now, one of my favorite quotes in this chapter is from David Altshuler, who basically said, you can cure something in a mouse or you can cure a disease in a few patients, but then when you actually want to make a medicine, it's like building a bridge with Lego and then having to build a bridge across the Hudson. And I think that's a great analogy because, as you mentioned before, for a true innovation to impact thousands or millions of patients around the globe, you need to do it at scale. And so just doing it in the laboratory is not enough. A medicine actually has to be then produced reliably, safely, and then be able to be distributed across the globe, which I think also people don't necessarily appreciate. We all appreciate that an iPhone is built and goes around the globe, but a medicine similarly has to be produced and then has even higher standards, right? Because it has to be the same everywhere and it has to be safe wherever it goes.
Right. And across lots of different populations, right? And the ability to deliver that to 150,000 people in Nigeria, as your book explains, right, is very different than maybe doing it here in New York City.
Right.
And I appreciate that quote that's in the book on 241, where, it says, it's a large engineering problem with expertise needed across cell biology, bioinformatics, molecular biology, and biophysics to make one of the most sophisticated medicines ever developed, right? I mean, it's an all hands on deck across industries experience.
Yeah.
Yeah.
And that can often take many, many years. So even though, CRISPR Vertex had treated their first patient, I forget exactly what year, in the mid 2000, I think it was 2015 or so, it still took 8-9 years for the medicine to get approved, because then you still have to show that it's effective in many, many patients. And then you have to have the manufacturing process in place and so on and so forth. So I think a lot of these parts of the breakthroughs are underappreciated by the general public.
You also mentioned in this chapter, the process is the product. And I think that's something that my listeners really understand, is that the way we do innovation is oftentimes just as valuable as the outcome to that innovation. And that's the part that gets replicated. That's the part that becomes the day-to-day of doing the work, of finding the outcome, and of creating the commercialized product that we could actually ship around the world.
Yeah, it's got to be reproducible for sure.
Yeah. It's just such a fascinating conversation. And I think, the reason why I love having guests from so many different fields on is that we learn from one another, just like this example, because, just as, Procter and Gamble is going from hot water tide to cold water tide, there's a lot of chemistry involved in that shift to get your clothes just as clean as it did in hot water as it is to cold. But I find it happening all the time, and really smart innovators lift their head up, look beyond their own microscope, beyond their own industry, and they begin to deploy open innovation. They begin to deploy different mechanisms outside of their own industry. One of my guests whose podcast dropped this week is a guy named Steve Rader, and he was the head of open innovation at NASA. Mm-hmm. And he enjoys telling this story of the potato chip conundrum. And that is, when you dig your hand into a bag of potato chips, the last thing you want to do is take your hand out and it's greasy and coated with salt and gross, right?
Mhm.
You want to joyfully eat your bag of potato chips without becoming a filthy mess by the end. And so this was a pervasive problem in the potato chip industry forever. They tried shaking it, they tried drying it, dehydrating it. It would change the flavor of the potato chip. Nothing worked, or it would break them. And so one potato chip company decided they were going to put out a query, but they didn't want their competitors to know. They wanted this to be their secret sauce. And so they camouflaged the query to say, how does one remove a viscous layer from a fragile disc, I think it was, a fragile wafer? And so in camouflaging that, they put the query out across lots of different industries to figure out who would come up with the best answer. It turned out it came from a violinist.
Oh,
And the violinist said, oh, I know how to do that. Every time I rosin my bow and play, it's actually the sound waves, it's sonic waves that remove the resin from my bow, and I have to reapply again. So this potato chip factory realized that with sonic waves, they could lift the final layer of oil off the chip, the extraneous oil, and then pass the chip on by removing it. And so you're never going to hire a violinist necessarily to work in your potato chip factory. But the
yeah.
answer is somebody has the answer, and it may come from the most unlikely source.
Yeah, totally agree. And so I think the lesson really is to have an open mind and then also give people the freedom to be able to think sometimes. Sometimes people on project teams are just do. But sometimes you also need to take a step back and have time to read other areas, attend lectures in other fields, and so on, because you never know where the next innovative breakthrough idea is going to come from.
Okay, so this brings us to your last chapter in the book, which is called How to Innovate: Insights from the Front Line of Drug Development. So can you give us some key takeaways, Dr. Powell, of what we should be thinking about as we go back to our own innovation cubby holes and get back to work on this? How else should we be thinking about how to innovate? What are the lessons that we can learn from drug development?
Yeah, I think, there's 10 lessons, so I probably won't go through all of them. One of the key ones for a medicine, though, and I don't know if people have thought a lot about this, is that for every medicine to actually get launched, it needs to get to the intersection of three circles in a Venn diagram. So one of the circles is biological understanding of the target. And again, target is what the medicine hits in order to make an effect and improve a disease, so to speak. So you need some biological understanding of that target. The second circle is the clinical understanding of where that target is relevant. And that means, what disease is that target relevant for, and in what state of that disease, and so on. Because if you don't know that, then you don't even know what disease to develop your medicine for. And then the third one is technological advancement in order to make the target druggable. So that target may have been previously undruggable or not drugged in a very good way, and somebody came up with some technological advancement to do it in a better way or to do it for the first time. So for a medicine to get approved, you really have to have all three of those come together. And I'm sure in other fields, there are similar or analogous types of Venn diagrams where you need to have two or three different elements come together in order to make the final product. The reason I highlight that is because then, sometimes just innovation won't work if you're too much in the biology and you're not really thinking about a disease, or if you are treating a patient with a disease, but you have no understanding of the disease and the biology of it, or you just don't have that technological breakthrough. The other reason I bring that up is because then you can really focus on what does it take to get to the middle of that Venn diagram. That's just one example of the innovation that's required in life sciences. But as I said, I think it would probably be analogous to other areas as well. And then, of course, there's the other elements such as persistence, resilience, and so on. I would ask people to read the book to get other insights.
Yeah, I think I was most taken with the better decision making too.
Mm.
Really stopping for a moment and examining our own biases, conscious or unconscious biases, certainly the blame game, right? Like our decision making is about who got the last one or who gets the next one. There's a lot of emotional work that goes into innovation. And we have to check ourselves
Yeah,
often.
for sure. I mean, I think one of the things I say in the book is, the best teams are the ones that all row in the same direction. It's fine to have disagreements, and in fact, it's good to have disagreements and differences of opinion because hopefully that refines the idea.
Right.
But once you make that decision, you should move forward. What I say is to have intellectual friction without social friction. You don't want to have the arguments go beyond that and then people making decisions based on grudges that they have rather than and then the data. The other most important element is to try to be as unbiased as possible. When you work on a project for years, even decades, you can get very wedded to the idea, you can get very wedded to what you think should be the outcome, but you do always have to consistently look at the data that's being generated and then say, does that fit my hypothesis or do I actually need to change my hypothesis? Or as we discussed about the killer experiment, do I need to really stop what I'm doing and think about a different approach?
Yeah.
That's very hard.
Excruciating. And I think everyone who's listening to this episode will know there comes a time when you have to kill your baby.
Yeah.
And the only thing that we have as a takeaway is, what did I learn? What did I save us by not pursuing this path?
Yeah.
And what does the learning generate for the next step? And, how is that this failure taking us forward from failure to forward is how I like to think about it.
Yeah, I mean, I think the measure of success is not necessarily whether you actually make it or not, but if you're making good decisions along the way and you're really being disciplined about that and focusing on doing your best and making the best decisions as you go along. I think the analogy there, this is not in the book, but the analogy there for me is tennis. I play a lot of tennis, and there's some people, who get very fixated on the score, very fixated on the outcome. I just want to win. But actually, what you need to do is focus on every shot. Are you hitting it in the right place? Are you serving well? Are you making those key decisions? And then the points will come in terms of winning. If you're only focused on the end game all the time, you can, one, drive yourself nuts. But then two is that then you become focused on the wrong things.
Mm. This has been such a fascinating discussion, Dr. Pao. I'm so glad we could sync up today on the podcast. Well, I'm going to ask you the 3 hot seat questions that I ask everybody who comes on the show. What is the greatest innovation of all time?
Oh, wow. The greatest innovation of all time?
All time.
Well, that's a tough one. I'm going to say, since we talked about it, I'll say the development of antibiotics.
Amen. Could not agree with you more. Okay. And not a lot of people say that on this show. I think I get more responses to fire than I do antibiotics.
Okay. But that really impacted life, as we said, in the past century.
For sure. If you had the opportunity to join any innovation team in the course of human history, which team would you have liked to have joined?
Yeah, I guess I would have to say then going to the moon. That's probably cliche, but it is one of those ones that you would think, how did we even, first of all, who even came up with the idea of doing that? And then how did we even do it? It is quite remarkable that we were able to do that. I think in 1969, right?
That's right. That's right. Who knows? Maybe Jeff Bezos will take you up on his next little flight, at least to the edge of the atmosphere. And my last question is, what is the innovation that the world needs now?
That's an interesting one.
Mm-hmm.
Well, I think it's how to make the planet sustainable such that everybody can live a satisfied life that is productive and impactful. That's a very generic answer, but I think as we enter this period, I think it's an important mindset that we're going to need for the future of humanity and the planet.
And on that note, Dr. Pao, if people want to get in touch with you, find out about your book, what is the best way for them to reach you?
Yeah, LinkedIn.
Fantastic. Dr. William Pao, thank you for joining me on the Innovation Storyteller Show today.
Thanks a lot, Susan. I really appreciate it.
Thank you.